So let's talk about mechanisms of microevolutionary change. Here's the syllabus objectives. There's 3, but this third one is about reviewing data or doing a simulation and reviewing data from that. So we're only going to talk about the first 2. Now, microevolution, the syllabus says that microevolution is small-scale variation in allele frequency within a species or even within a population. So it means that there's actually no new speciation. It's within the same species, small-scale variation within a species. Okay, so there's actually a number of mechanisms of microevolution. Firstly, mutations. We've previously said that mutation is really the only way in which we get new alleles into a species. So that's really important. Another way in which we get new alleles into a population is through gene flow. Gene flow is immigration and emigration of organisms into a population. Both of these introduce new alleles, so they're beneficial because they increase variation. The next one we're going to talk about is called genetic drift, and this is like random chance events that actually reduce the number of alleles by wiping out a proportion of the population in a species. So the risk there is that there's a decrease in variation because it's a removal of alleles. Again, all 3 of these processes are random, but natural selection is non-random. It is, as we said in the last video, natural selection is a change in allele frequency in response to a selection pressure. So when we talk about mutations, we're talking about genetic mutations, a change to the genetic code, which creates new alleles and variation. It's a random process. It may be beneficial because it might introduce new phenotypes that provide a selective advantage, a reproductive and a survival advantage. But they may also introduce disease. Mutations are genetic disorders. Gene flow also is a random process, and it's when we have movement of alleles in and out of a gene pool. This occurs through immigration and emigration. It could be beneficial in several ways. Immigration might increase favourable alleles into a population. Emigration might be beneficial because unfavourable alleles might actually leave the population. So gene flow is generally beneficial and it increases the variation. And what we've said in the previous video is variation or a large gene pool is important for survival because it allows a species to be able to adapt to changes in their environment. Now, genetic drift is where we have a change in the frequency of existing alleles due to random chance. And now, so you imagine it might be something like a natural disaster, the introduction of a predator might be hunting, for example. And what we see is we get a reduction in the size of the gene pool. And so that has a negative impact because there's a loss of variation. There's 2 specific examples that we're going to talk about, bottleneck and the founder effect. So bottleneck is where we have some sort of event that greatly reduces the population size and therefore the genetic diversity. For example, a hunting of a species can greatly reduce the population and therefore greatly reduce the gene pool. So as the population increases again, if it recovers and increases, well, it's recovering from that low gene pool. So there's a less variation and they're more susceptible to changes in the environment. Overhunting, natural disasters such as volcanoes, fires and earthquakes, all of these can greatly reduce a species population and therefore greatly reduce the gene pool. Now, the founder effect is where we have a small number of organisms from a species moving to a new area and colonizing a new area. So they only take the alleles that they've got within those organisms to that new area, and they start to populate and colonize an area only with those alleles. So again, we've got a limited gene pool. So whilst those previous mechanisms of microevolution, mutations, gene flow, and genetic drift, we're talking about random mechanisms of microevolution. Now we're going to think back again to natural selection, which is non-random. It's a change in allele frequency in response to specific selection pressures. So, as I've talked about with variation, we see that there's a wide range in variation in phenotype, and it's distributed with a normal distribution, with most organisms demonstrating an intermediate phenotype. Now, we have selection pressures that can alter this normal distribution. Let's have a look at those. Firstly, stabilising. So a stabilising selection is where we have selection against the extremes. So we end up having more of the population in that middle area and a reduction in the extremes. A great example of this is birth weight for human birth weight. So if a baby is too small, then its chances of survival decrease because it's just too small to survive. If it's too big, it's very, very difficult to get through the birth canal without damage. So we find that there is a stabilising effect where the majority of the baby's birth weights are stabilised around the middle, and there's selection against the extremely low and extremely high birth weight. So that's an example of stabilising selection. Directional selection occurs when one of the extremes is selected against and one is selected for. So we see change in one direction. The frequency of one allele increases and the other decreases. An example of that is the peppered moth. So when there was the change in the environment such that the trees became darker in colour, well, the white moths were selected against and the darker coloured moths were selected for. So we saw a change in allele frequency moving towards that dark side. So that's directional selection. And so, but there is also a third example. So let's have a look at these. So let's have a look at these squirrels. You can see that one of the squirrels has a very long tail and one has a short tail. We can also see that the long-tailed squirrel is on a tree and the short-tailed squirrel is on the ground. So they essentially occupy different niches. Whilst they're the same species, we see this significant variation. And the squirrels that live in the trees are the ones that have the long tails for balance, and the ones who live on the ground have a short tail for speed. So we see a disruptive selection that favours the extremes because they occupy different niches. We have an intermediate phenotypes that get selected against. So these intermediate ones, the ones in the middle, get selected against. And we have selection of the extremes. This occurs when we've got unstable environments. This occurs when we've got, when they occupy slightly different niches. So that's an example of disruptive selection. So we have stabilising, directional, disruptive. Stabilising, directional, disruptive.